So I get to introduce Stella today. She joined LAAS in the labs of mine and Dr. Jake Jungers on a Monsanto Bayer Fellowship in 2018. And she actually, pretty quickly landed on this idea of doing isotope labeling work for a lot of her, for two of the three chapters that you'll hear about today. And what was really amazing, pretty quickly on, was that I was walking through lab one day and Stella was like, "Oh, look at what I made." And what she made was basically everything that she would need. So she built her own CO2 and temperature and humidity monitoring sensors. She built, and with Jake and I constructed and designed this whole revamped labeling chamber from the very defunct thing I had inherited. She developed the dehumidifier, the methods, kind of everything that she would need. And we even were at the point of wondering if we could hire her to make a cottage industry as another source of funding for the labs so that we wouldn't have to work on grants as much. And it was just I was whatever you need. Do you need a soldering iron? No, I actually already have one at home because I enjoy doing this on the side for fun, and found out that her and her husband are actually reconstructing a whole trailer to go camping and doing things on the weekends. And so it was a lot of fun to get to see Stella in action with all of this creation and building, as she is also constructing, these quite nice, elegant research questions. She's also been I also want to give a shout out to what a great leader she's been in our graduate program. She has explored ways for the grad students to get into more K through 12 education. So she served for quite a long time on our grad student program organization. She's been the chair and co-chair of the Agronomy Society Perennial Grains Community. One thing that to me was really special is that she ran my IRMS lab for some time when the lab manager was traveling. And during that time, she wrote new code so that we could better process all of the data coming out of the lab. And I think what was really remarkable to me is that the humor and grace and kindness that you had in teaching the lab manager then how to use all of that code, because this is a person who was absolutely terrified of even any kind of data processing, even without anything fancy involved. And so I really appreciated and was really impressed by your ability to teach other people who could be kind of terrified of some of the processes that we do. And so the last picture I actually want to show is of the day that we were getting this labeling set up. And even in the midst of pandemic with all of these huge meter cubed labeling chambers, you still had a smile on your face. And I think that is what we will definitely miss as we see you off into your next chapters. So with that, we'll now hear from Stella for your three chapters, isotope labeling and root decomposition. So take it away.
All right. Well, thank you, Jess, for that really awesome introduction. I'm going to get this set up here. Okay, and could I just get a thumbs up from someone that you're seeing the regular slides and not the slides with notes? Looks good. All right. Yeah, well, thank you again, Jess, for that introduction. It was a great, I've had a great time working in both yours and Jake's labs over the last few years, and I'm really excited to share with you all some of the stuff that I've been working on. So with that, I'm going to talk about my thesis research, which was focused on evaluating the physiological and stand age-related controls on soil carbon and nitrogen dynamics in perennial and annual grain systems. And so with this, the general outline of the presentation, I'll go through some kind of background introductory material, and then I'll get into each of my thesis chapters and the studies that are related to those. And then I'll finish off by trying to do some synthesis and concluding remarks to connect these different studies together. So moving into the introduction, over the past millennium, the percent of global ice-free land that's been in agricultural production has increased from about 4% to 40%. With this transition from native landscapes into cultivated landscapes, about a third of that global ice-free land is specifically used for cropland production. And within this cropland, most of that land is used to produce annual grain crops. As an example, in the United States, croplands are dominated by just 3 annual grain species. So corn and soybean alone account for about 56% of the cropland acreage in the United States. And then wheat, being the third most produced crop, accounts for about 10% of that cropland acreage in the United States. So together, these just 3 annual grain crops are accounting for almost 70% of that cropland acreage. And the conversion of native landscapes, which, contain a higher diversity of plant species and different cycles that are associated with that. This conversion from a more diverse system to one that is dominated by annual grain monocultures marks a significant decline in the diversity of plants occupying these landscapes, and it kind of serves to threaten the economic, cultural, and environmental sustainability of our agricultural systems. So this kind of leading off on that environmental sustainability piece, this long-term annual crop production often leads to soil erosion, losses of soil carbon, reduced crop productivity, and then greater nitrogen contributions to water and air pollution. These have some pretty tangible economic disadvantages. So with those losses in soil carbon, in some parts of the Midwest, we see that that's associated with a 6% decline in yield, which amounts to about a $3 billion loss in the economic value of that land. Similarly, with our nitrogen, when we're losing more nitrogen from these systems and polluting some of the groundwater resources that societies rely on, in some states across the Midwest, they're spending about $5 billion annually to treat these groundwaters that have been polluted by agriculturally derived nitrogen. So in order to preserve the long-term productivity of soils and reduce these economic losses, we really need to be focusing on developing cropping systems that better retain that soil carbon and nitrogen in the system. And with my research, we kind of focused on perennial crops as a strategy to retain the carbon in the system, better regulate that agricultural nitrogen usage, and start to preserve some of that economic and environmental sustainability of our agricultural systems. In particular, these perennials are pretty well suited to meet these goals because of some unique physiology that differentiates them from annual crops. So in particular, we focused on root growth and turnover. Those 2 processes largely control soil carbon accrual. And in our perennial cropping systems, we tend to see that they have higher standing rootstocks, greater root growth and decomposition, and therefore a greater capacity for carbon and nitrogen cycling. We also can look at the carbon uptake and partitioning patterns. So this carbon uptake and below ground carbon inputs similarly are largely linked to soil carbon dynamics. And we tend to see with our perennials that we have higher carbon uptake. A larger proportion of that carbon that's assimilated is allocated below ground. And then within that below ground pool, we tend to see that there's a higher allocation to the soil microbial communities, which, as we're kind of beginning to understand, that movement of below ground carbon from roots into soil microbial communities is a pretty good precursor for the ability of that below ground carbon to become stabilized. And then we can also look at the nitrogen side. So thinking about how these perennial crops can source nitrogen and conserve it once they've taken it into their tissues. So perennial crops are pretty efficient at taking up nitrogen that's in the soil. And then they have these really well-developed conservation strategies to keep that nitrogen moving between above and below ground tissues to reduce those nitrogen losses. Together, this kind of makes these systems have a reduced reliance on fertilizer nitrogen because of that efficient fertilizer uptake and those well-developed conservation strategies. And when I talk about nitrogen conservation, I'm really talking about the efficiency that nitrogen is translocated internally in these crops from above ground tissues to below ground tissues. This is usually something that happens between maturity and senescence. So as these perennial grass crops approach maturity, they start taking the nitrogen that's stored in their above ground tissues and put it into below ground storage organs. This internally translocated nitrogen can account for up to 70% of the nitrogen used to support new tissue growth during the subsequent growing season. And so it represents a pretty important and significant source of nitrogen to meet the crop's nitrogen demands. And so altogether, these physiological traits that I've identified can all enhance that soil carbon and nitrogen cycling in agricultural croplands. When we think about perennial systems, there's already a variety of perennial cropping systems under development that could serve the purpose of building soil carbon and regulating agricultural nitrogen, including perennial biofuel crops, perennial forages, and then emerging perennial grain and forage systems. And these perennial grain and forage crops are particularly exciting because they have the potential to provide not only food for animals through forage, but also food for humans through that grain production. And they're undergoing sustainable development through the environmental side up through the economic and supply chain side, which makes them a pretty exciting alternative to annual grains. Of these perennial grain and forage crops, intermediate wheatgrass is one of them, and this is the crop that I focused on in my research. This is a cool season perennial grass. Again, it's domesticated for both grain and forage production, and when harvested or when produced for grain, it's harvested for grain at maturity. It's already been shown to provide a lot of ecosystem services, including protection from erosion, improved soil aggregation, reduced nitrate leaching, and some evidence that intermediate wheatgrass can build more robust soil microbial communities and potentially contribute to soil carbon accrual. So coming back to these traits that I identified as being important for regulating soil carbon and nitrogen in agricultural systems, intermediate wheatgrass already demonstrates some of these traits. So we know that when compared to other annual grains, intermediate wheatgrass has larger standing rootstocks. The photosynthetic activity and subsequent carbon uptake tends to be greater in intermediate wheatgrass than annual counterparts. And there's a few studies that have also started to show that this fertilizer nitrogen uptake is pretty efficient in our intermediate wheatgrass crops when compared to other annual grains such as wheat. But there's other traits that might be impacted by our intermediate wheatgrass management practices, in particular those relating to nitrogen conservation. So in our perennial biofuel systems, we tend to see that management recommendations recommend harvesting that biomass at senescence in order to allow those crops to carry out that nitrogen conservation. So moving that tissue from above ground to below ground pools. But in intermediate wheatgrass, we harvest it at maturity for grain, which is potentially cutting off this ability of the crop to conserve nitrogen and could lead to some sort of nitrogen limited status as these stands age. So what we were really interested in with my research was evaluating whether or not the rest of these traits were expressed in intermediate wheatgrass and how consistently they were expressed across stand age. And then, as I kind of mentioned just now, that piece about how our management is affecting the ability of intermediate wheatgrass to carry out some of these physiological traits that might be advantageous for soil carbon and nitrogen regulation. All right, so that brings me to my thesis research, where we're looking again at that physiological and stand age related controls over soil carbon and nitrogen dynamics. The first study, we were focused on that root growth and decomposition piece. The second study kind of spearheaded that carbon uptake and partitioning. And then our third study will get into the nitrogen sources and nitrogen conservation employed by intermediate wheatgrass. So we'll start by looking at this first study with root growth and decomposition. In this study, we were mostly interested in looking at changes in standing rootstocks, root growth and root decomposition as they occurred between perennial and annual grains, and then assessing those same root parameters as they occurred across intermediate wheatgrass stand age. And again, as I outlined previously, we were expecting that our perennial grains would have greater standing rootstocks, a greater capacity for growth and decomposition, and therefore a greater capacity for carbon and nitrogen recycling between root tissues and soil pools. This study was conducted in Rosemount, Minnesota in 2020 and 2021. We were looking at 3 cropping systems. So spring wheat was representative of our annual grain, and then we had the 2 intermediate wheatgrass systems, a first year and a second year system. And we, within each growing season, sampled the cropping systems at 5 different growth stages. Today, we'll be looking at kind of seasonal trends in standing rootstock, and then we'll look at the total new root production, root decomposition, and root carbon and nitrogen turnover that occurred over the whole growing season. So to evaluate these parameters, we used paired sequential core and root ingrowth core techniques. These sequential cores were representing our standing rootstock. So for this, we just extracted a core of soil to a vertical depth of 15 centimeters at a 45-degree angle to the soil surface. In that same cored hole, we then inserted a mesh cylinder that we filled with root-free soil, and this was representative of our root ingrowth core. We left these cores in situ for about 6 weeks, which allowed those crops to recolonize the core, but prevented a lot of decomposition from happening. And so the roots that we captured in these cores were representative of new root production or gross root production. So what this kind of looks like on a timeline, we installed 4 of these root and growth cores across the growing season. We would install 1, allow it to sit for 6 weeks, come back, extract it, and on that same day, we would install another ingrowth core. And then, of course, at the same time, we took our sequential cores for measuring our standing rootstock. Each of these sampling events aligned with a growth stage. So we were looking at root growth dynamics between the vegetative, elongation, physiological maturity. And then in our perennial systems, we also looked at the early regrowth and late fall regrowth stages. And then today we'll be looking at new root production. So we're just taking the total, the cumulative root mass collected from each of our ingrowth cores, and then we'll look at the decomposed root biomass across the growing season. And this was determined by taking the difference in the first and the final sequential core sampling and subtracting away our new root production, which gives us an estimate of our decomposed root biomass. So we'll start by looking at this standing stock data set. And what we can see first here is that that second year intermediate wheatgrass field tended to maintain its standing stock across the study period or the growing season. Comparatively, we can see that our first year system tended to build root biomass across the growing season, which is in line with how we think about these systems as, they're planted and then they're really rapidly trying to acquire resources and establish themselves. And in line with numerous other studies, we saw that our intermediate wheatgrass systems both tended to maintain higher root stocks than wheat. And the second year field had a somewhat larger root stock than our first year field. So in line with this, when we look at our total root production and root decomposition, our root production and decomposition were both greater in our intermediate wheatgrass systems than in wheat. But across stand ages, we start to see that our first year system had higher root production and lower root decomposition, whereas in our second year system, we saw the opposite trend where our root decomposition was greater than our root production. So this marks a shift in our system where we're seeing the first year field dominated by root growth, the second year field dominated by root decomposition. And consequently, then this kind of drives these patterns in root carbon and nitrogen turnover, where we see that our perennial systems, again, have greater carbon and nitrogen turnover than our annual wheat system. So some of the key points that we take away from this study, again, our root growth and decomposition was greater in our intermediate wheatgrass system than wheat, suggesting that these systems have a higher capacity for recycling that carbon and nitrogen that moves into the root system. We also saw a pretty clear age-related shift in intermediate wheatgrass root dynamics. So we transitioned from that system dominated by root growth in our first year field to one that's dominated by root decomposition in our second year field. This shift in root growth and the decomposition processes has some implications for the potential for soil carbon accrual. So we tend to think about root growth as being associated with root exudation and the input of these kind of low molecular weight substances into the soil pool. And these inputs related to exudation tend to be more preferentially or easily stabilized than those that are derived from the turnover of root tissue. And so having this shift from root growth to decomposition, we might expect then that the potential for that root carbon entering the soil between our first and second year intermediate wheatgrass production year might decline as those stands age. But, in this study, we were just looking at root growth and decomposition processes, and we didn't actually get to see where that root-derived carbon goes. So this kind of leads into our second study where we were looking at directly tracing the movement of carbon from above ground tissues into below ground soil pools. In this study, we were hoping to quantify the carbon uptake and below ground carbon partitioning in perennial and annual grains. And again, as with our first study, we were interested in looking at these same parameters, but within intermediate wheatgrass stand age and not just between perennial and annual grains. Again, kind of a reminder of what we talked about earlier. We're expecting that the intermediate wheatgrass perennial systems will have a higher carbon uptake, a higher below ground carbon allocation, and then a higher allocation to soil microbial communities. Similarly, this study was conducted in Rosemount, Minnesota. We conducted a study in 2020. We used the same cropping systems. So we had a spring wheat, a first year intermediate wheatgrass, and a second year intermediate wheatgrass. And we used pulse chase labeling methods for this study, where we were adding a tracer and then we sampled 10 times over a 90-day period after that tracer was added. So these methods involve, as I mentioned, this carbon isotope tracer methodology. For these methods, we put this chamber over the soil surface and spike that chamber atmosphere with 13 carbon. The plants then naturally take up that tracer and start to assimilate it to plant roots, soil microbial communities, and then some of it gets lost through respiration. We added our tracer in mid-May to 4 replicate areas in each field. The labeled areas were then sampled 10 times over the subsequent 90 days, which you can see on the timeline in the figure on the bottom there. At each sampling event, we sampled shoots, roots, soil, and soil respiration. And then for the first 6 sampling events extending through 7 days post-label application, we also sampled rhizosphere PLFAs, which are an indicator of the soil microbial community. And all of our root and soil pools were sampled to a depth of 15 centimeters. So we'll be looking at 3 main parameters here, and we'll start with the total new carbon uptake and partitioning. This is the absolute amount of tracer carbon that we recovered from each system, and it's just looking at the mass of carbon that we recovered in milligrams, 13 carbon per meter squared. We'll also look at the proportional carbon recovery, and so in this we're looking at within each system, what % of the total assimilated carbon was recovered from that compartment. So to get this metric, we're looking at the 13 carbon that we recovered at a sampling event and dividing that by the 13 carbon recovered at peak tracer recovery. And peak tracer recovery typically occurs within the first 0 to 1.5 hours after tracer addition. And then finally, we'll look at the proportion of new microbial biomass carbon recovered from microbial functional groups. And for this, it's kind of similar to the proportional new carbon recovery, but we're just looking at the microbial biomass. So we'll take the 13 carbon recovered from a given functional group and divide it by the total microbial biomass, 13 carbon. And because all these the shoots, roots, soil, and soil respiration pools, we're kind of interested in seeing how carbon moves through each of those within a given cropping system. We'll kind of go through this data one cropping system at a time, and then do some larger comparisons throughout. So we'll start by looking at wheat. And our first takeaway is that the wheat carbon uptake was relatively low, which we can see in the red outlined area, but the carbon that it did take in tended to be retained in the soil. And we can see this again when looking at the proportional data. So despite that relatively low carbon uptake, much of the carbon that was taken into wheat was allocated to soil, with up to 50% almost of that carbon assimilated being retained in soil pools. Indicating that this wheat system rapidly processes the carbon inputs that it receives. We can also see that the proportion of new carbon in soil microbial communities was relatively high in our wheat system. So not only did wheat effectively allocate or translocate carbon from tissues into below ground soil pools, these soil microbial communities were also pretty efficiently being able to access that carbon. And since the inclusion of new carbon in soil microbial communities is an important pathway by which soil carbon is stabilized, this suggests that wheat might have a little bit of an advantage for moving carbon that it assimilates into more stable soil carbon pools. And so to summarize, we have low carbon uptake in our wheat. Most of that carbon that is taken in, though, in terms of below ground partitioning, was stored in soil. And we actually saw that a pretty high amount of that, or relatively high amount of that carbon then was retained or recovered from microbial communities. So this kind of leads to wheat as being able to contribute larger, more localized soil carbon inputs over a shorter term. So now we can look at our first year intermediate wheatgrass system, where we see a pretty high carbon uptake, but a relatively lower retention of that carbon below ground. And another important thing to note here is that if we look at the tops of these bars, that carbon that we recovered initially compared to the amount of carbon that we were recovering by the end of our study period, we were missing about 30% of the carbon that we initially recovered, suggesting that that first year intermediate wheatgrass field was moving carbon into a pool that we did not sample. And so in contrast to our wheat system, we see that the carbon that the first year intermediate wheatgrass field did assimilate tended to be stored in roots rather than soil. This is in line with our first study, where we saw that the first year field was dominated by root growth and resource acquisition. And it's a pretty important finding because most studies measure and compare soil carbon between perennial and annual grains at a single point in time, but they don't take into account any kind of longer term contributions that stored carbon might be making. We know that storage of carbon in roots can be important for kind of extending that residence time of assimilated carbon in soils. And so the contributions that intermediate wheatgrass is making to soil carbon might be temporally extended by storing this carbon in roots. In addition to having this potentially longer, wider temporal effect, we also found evidence that there might be a wider spatial effect of intermediate wheatgrass on soil carbon contributions. We know that intermediate wheatgrass tends to support the development of arbuscular mycorrhizal fungi networks. And in this study, we found that that first year field invested a relatively larger proportion of microbial carbon into those arbuscular mycorrhizal fungal networks. This then could serve to kind of extend that spatial footprint of the carbon inputs that the host intermediate wheatgrass plant is making into the soil. And by moving that carbon out throughout these arbuscular mycorrhizal networks, could be extending that carbon input, pretty far beyond where our host plant was. And this could in part then explain some of that missing 30% of carbon that we did not recover that I mentioned in the previous slides. And again, this just kind of underscores the need to account for longer term contributions that these intermediate wheatgrass systems might be making to soil carbon when we're making comparisons between our perennial grain and annual grain cropping systems. So in summary, we see that our carbon uptake in the first year intermediate wheatgrass field was relatively high. Instead of storing that carbon in soil, we stored it in roots in our first year field. The allocation to microbial communities was low, but the carbon that was recovered from microbial communities was mostly stored in those arbuscular mycorrhizal networks. And this is all indicative of a system that's characterized by acquisitive growth. And because of that storage in roots and arbuscular mycorrhizal networks, we could expect that intermediate wheatgrass is making possibly smaller but more widespread contributions to soil carbon over a longer term. And finally, we'll look at our second year system, where we see, compared to our first year system, a pretty dramatic decline in the carbon uptake. And we can also look, between all of our systems and see that that second year field is storing really low amounts of that new carbon in below ground pools. Instead of storing that carbon in below ground pools, most of that new carbon is actually just being retained in above ground tissues. So by the end of our study period, there was still about 50% of that new carbon just lingering in the above ground tissues. And we also see that a pretty high proportion of new carbon was respired or just lost from the system through soil respiration. And together, these properties or these results suggest that the second year intermediate wheatgrass system is transitioning into a phase of more conservative growth, characterized by investment in above ground tissues and decomposition. We also see that the second year field stored a relatively high proportion of new microbial carbon in saprotrophic fungi, which are generally associated with the decomposition of organic matter inputs. Coupled with the high loss of new carbon to respiration, these results again support findings from our first study, where that second year system is transitioning into a phase that's dominated by the decomposition of below ground carbon inputs. And this study kind of takes it one step further and shows that, from our first study, we saw that second year system had a lot of root decomposition, and we suggested that could have some advantage for recycling carbon into the soil system. But this study suggests that increase in decomposition isn't actually leading to any kind of advantages for below ground soil carbon accrual. And so to kind of put the last piece of the puzzle in here, we see that our second year field had a moderate carbon uptake kind of in between wheat and our first year system. But the partitioning of carbon below ground was pretty limited. We didn't see a lot of that carbon going into microbial communities, and the carbon that was moving into microbial communities was mostly accounted for by saprotrophs. And altogether, this suggests that the second year system is making pretty limited contributions to soil carbon. It's transitioned rather into a phase of conservative growth that's dominated by high decomposition and high new carbon losses from the system. So just to kind of summarize some of these key points, again, when we're thinking about the potential for intermediate wheatgrass to sequester carbon or build soil carbon, and we're making those comparisons between intermediate wheatgrass and wheat, we really need to be considering the long-term contributions of intermediate wheatgrass to soil carbon through that longer-term decomposition of intermediate wheatgrass roots. We also saw that there is a shift from an acquisitive growth strategy in our first year system to a more conservative growth strategy in our second year system. And again, this aligns well with our results from study one. And the increase in decomposition that we saw in study one in our intermediate wheatgrass systems, this study kind of builds on those results and suggests that decomposition is not leading to enhanced soil carbon retention or soil carbon inputs. And so we can start to wonder and think about what's driving this shift in carbon cycling and what's driving this limited carbon retention in our systems. And we know that paired nutrient dynamics, especially nitrogen, can limit the potential for soil carbon accrual in cropping systems. And so that kind of leads us into this third and final study where we were looking at nitrogen sources and nitrogen availability in our intermediate wheatgrass systems. For this study, we were looking at the assessing the fertilizer uptake efficiency and indicators of nitrogen availability and limitation in intermediate wheatgrass. We wanted to quantify the primary nitrogen sources used to support tissue growth in intermediate wheatgrass, which could help us understand the reliance on nitrogen fertilizer. And then we evaluated proxies of nitrogen conservation to assess the development and reliance on translocated nitrogen for meeting nitrogen demands. And again, just going back to that kind of first piece in the introduction, we're expecting that our intermediate wheatgrass systems will have a pretty well-developed nitrogen conservation. We'll be really good at taking up fertilizer nitrogen, and we'll have a high nitrogen availability, and that together these attributes will reduce the reliance on fertilizer nitrogen. So for this study, we were again conducting it in Rosemount, Minnesota in 2021. Here we were just looking at intermediate wheatgrass. So we had a first year, a second year, and a third year system. And as with our other studies, we were doing in-season sampling that aligned with some key growth stages. And for this study, we used a 15 nitrogen enriched urea to trace some of those fertilizer nitrogen dynamics and nitrogen sources. And then we also had kind of a split plot, harvested and unharvested sampling plots to assess the nitrogen conservation strategies employed by intermediate wheatgrass. So our timeline for this study, crops of course start growing in the early part of the season, and we applied our 15 nitrogen urea in mid-April, along with the paired plots, which were to which we did not apply labeled urea, but unlabeled urea. And in both of those plots, we applied that fertilizer at a rate of 80 kilograms of nitrogen per hectare. Crops were allowed to grow throughout the season and uptake that fertilizer tracer. As I mentioned, we kind of had this split where some of the plots were harvested and some were unharvested. As I talked about earlier, this is important for measuring nitrogen conservation because that nitrogen conservation and translocation from above ground to below ground tissues occurs between maturity and senescence. So of course, our harvesting disrupts that process. So we had some plots that were harvested as normal, but then we had some plots that we left unharvested to evaluate that nitrogen conservation. We then sampled at 4 different times throughout the season. So at boot stage, harvest, regrowth, and crop senescence. And today we'll just be looking at data from the samples collected at maturity, and then those samples collected at the end of the season from our unharvested system to look at fertilizer nitrogen recovery, nitrogen sources, and nitrogen conservation. At each sampling, we collected above ground tissues, root biomass, and soil samples. And again, for today, we won't look at all of these parameters. We'll just be looking at the samples we collected from our above ground tissues and the soil samples we collected to analyze soil available nitrate. And so here we'll get into some of the calculations and definitions of the variables that we'll look at. I'll try and explain these, but might go through them just a little bit quickly for time's sake, and I'm happy to discuss them further later. But so we'll first start looking at the fertilizer recovery efficiency, and this is at maturity. This is the percent of total applied fertilizer that was recovered from above ground tissues. We'll then move into looking at the percent nitrogen derived from our different sources. So we'll look at translocated nitrogen, nitrogen derived from fertilizer, and nitrogen derived from soil. The translocated nitrogen is the percent of above ground tissue nitrogen that could have been sourced from nitrogen translocated below ground during a previous growing season. Fertilizer nitrogen was measured from our isotopic samples and are labeled fertilizer, and it represents the percent of tissue nitrogen sourced from urea fertilizers. And then the soil nitrogen is kind of the remaining percent of above ground tissue nitrogen that didn't come from fertilizer or translocation, and it's assumed to be sourced from soil. And then we'll look at some of our proxies for nitrogen conservation, and we'll mostly just look at that line 6, our nitrogen resorption proficiency, and this is represented by the tissue nitrogen content at senescence, which represents the lowest tissue N content that the crop achieves throughout the season. All right, so looking at our fertilizer recovery efficiency, the general trend that we see here is that as our intermediate wheatgrass stands aged, we saw a larger proportion of applied fertilizer nitrogen was assimilated into above ground tissues. While this could indicate that the crops have a greater uptake capacity of fertilizer N, which is generally good, it's interesting too to think about why these trends are happening and what could be driving that increase in fertilizer recovery. And so we also looked at some indicators of nitrogen availability, which showed that as stands aged, nitrogen became less available. So we see that the soil available nitrate declined in our upper layer, declined significantly with stand age, as did our above ground tissue nitrogen content. And this is in line with other studies that have shown that as intermediate wheatgrass stands age, the net nitrogen immobilization occurs in both intermediate wheatgrass stands and other perennial biofuels. And so this increase in fertilizer uptake efficiency could therefore be driven by a depletion of nitrogen reserves in the soil and a greater reliance on fertilizer nitrogen to meet nitrogen demands. We see this echoed when looking at the proportional recovery of nitrogen in above ground tissues. The proportion of above ground tissue nitrogen sourced from soil declined across stand age, while the proportion of nitrogen sourced from fertilizer and translocated nitrogen increased. As nitrogen conservation tends to increase when nitrogen becomes more limited, this further supports that the system's nitrogen availability was declining as stands aged. And then we have one final piece of evidence that supports this development of nitrogen limitation and increased reliance on nitrogen conservation in our nitrogen resorption proficiency, where we see that this declining nitrogen resorption proficiency indicates that those plants are engaging in greater nitrogen conservation and potentially relying more on that nitrogen conservation to meet internal nitrogen demands. So to summarize this study, we saw that fertilizer recovery efficiency increased with stand age, as did the investment in nitrogen conservation, but we had a declining nitrogen availability. And so we suggested that as intermediate wheatgrass stands age, they become more limited in nitrogen and more reliant on fertilizer N. And kind of altogether, these changes in the system nitrogen availability could be driving those changes in growth strategy and potential for soil carbon accrual. All right, so kind of finally here, we'll try and put all these three studies together because they were pretty complementary to one another. And we see that from our first study. Again, we generally saw that we had greater rootstocks that increased across stand age. Our root growth declined, while our root decomposition increased with stand age. In line with this, we saw that in our second study, we saw that as intermediate wheatgrass stands aged, carbon uptake declined, as did our below ground carbon allocation, which again is in line with our first study where we're seeing this shift from growth to decomposition. And in our last study, we saw that some evidence of decreasing nitrogen availability going along with these increased reliance on fertilizer and internally translocated nitrogen. Since we know that that decline in nitrogen availability rather, can limit soil carbon accrual and could be driving some of these other shifts in growth strategy from acquisitive to conservative and such, we suggested that from all of this, kind of, I guess, the one main recommendation is that optimizing that nitrogen management is going to be critical for maximizing the potential for soil carbon accrual. And this nitrogen management also represents, on this list of things, one of the easier things for us to manage in this intermediate wheatgrass system. All right, and with that, I'll go ahead and acknowledge my committee, of course, Jess Gutknecht, Jake Jungers, Anna Cates, and Sarah Hobby, who have all been greatly helpful and great supports throughout this research. All of the members and technicians and students of the Sustainable Cropping Systems and Seekers Labs, and then our funding sources down there at the bottom. And yeah, with that, I will take any questions.
Wonderful. Thank you, Stella. Let's give Stella a round of applause virtually. So feel free to raise your hand. I'm gonna change my view here so I can see people when they raise hands, and we can start taking some questions. Also, if you would rather put your questions in the chat, feel free to do that. I'll help facilitate this process here. Alex.
Nice job, Stella. I was just curious if you had speculations on whether that missing carbon that you weren't able to measure might have been moved like laterally or like with depth.
Yeah, that is a good question, and it's definitely one of the limitations, I think, of the study, is that we can't exactly say whether that spatial extent that I talked about is lateral or moving up and down. I think in reality, it's probably a bit of both. So, we only sampled to 15 centimeters, and we know that, especially in that first year, those systems are putting on a lot of root mass and probably putting a lot of new carbon into roots that might be extending below that 15 centimeter mark. Most of the kind of a little bit of an in line with that, most of the root mass that these systems put on does tend to be above 20 centimeters so from 0 to 20, but there still might be some carbon moving down below that 0 to 15 centimeter depth. And then, I guess in terms of the lateral movement, when we think about intermediate wheatgrass root systems, it does kind of move laterally a lot and then a little bit below. So from our results, we can't quite disentangle or conclude where exactly in the system that extra carbon might be moving, but it is probably moving outside of that 1 meter squared area that we sampled in some fashion.
More questions? Dr. Mola.
Hey, thank you, Stella. I enjoyed your talk. Just one big picture question and then maybe a follow-up. How much carbon in tons per hectare does intermediate wheatgrass sequester or fix over the 2 to 3 years that you studied this process?
Yeah, that's a really good question, and I don't know the exact values off the top of my head of what amount of carbon it's sequestering. Yeah, I don't know, I can't recall off the top of my head, but
If you say it's 300 milligrams per square meter or something like that, what is that in tons per hectare?
yeah, that's again a good question. I'm not sure the conversion on the moving from milligrams per meter squared to tons per hectare. And it's a little,
I just work it out. I mean, what's a milligram to a ton or a kilogram? What's a square meter to a hectare?
yeah, I suppose I can stop sharing here, and I don't know those conversions off the top of my head, but I can look at them here. But I think sorry, let me see here. Oops. Sorry, Dr. Merlo, I'm having a little bit of trouble here.
No problem. Take your time. That sometimes when we do plot scale studies, the units aren't easily translated into something that can be easily interpreted relative to studies that are being done more at a global scale.
Right, and it's so it looks like the conversion is like 1 ton per hectare to about 100,000 milligrams per meter squared. So I guess in this study, we were kind of to that point. I mean, we were mostly interested in comparing the systems to one another, which is why we chose these units that are a little bit smaller and perhaps easier to work with than scaling it up to tons per hectare. And it, again, kind of aligns with what we've seen in the literature. So it made comparisons across, our studies to other literature values a little bit more easy. But yeah, I do agree that kind of scaling it up and thinking about the more global applications, I suppose, and using units that are reflective of that would be beneficial. I guess kind of to your wider question of thinking about this study as a metric of how much carbon intermediate wheatgrass systems can sequester. It's another, it's an interesting question that I don't know that this study totally gets at, I guess, just because we measured kind of in one growing season, and that carbon is pretty fluid and probably still moving around between these different pools beyond our study period. So in terms of kind of how this study contributes to our knowledge of how much carbon intermediate wheatgrass is sequestering, it's a little bit, I think, of a maybe stickier question that I don't know that our data can totally get at.
Just one follow-up. For your first year study, do you think the wire mesh cores that you installed had any effect on the root growth? In other words, would root growth into those sort of wire mesh cores in some ways be different from what's happening with the rest of the plant, or would you think it's representative of what's happening in the rest of the plant?
So I think that it is representative of what's happening in the rest of the plant. So there have actually been quite a few studies that have looked at, in the context of installing root and growth cores, what is the effect of severing those roots and installing the core? Is the subsequent root growth accelerated compared to normal root growth? Does it decline because you've kind of injured that root? But a lot of these studies conclude that, if you had an ingrowth period of maybe 1 week, it's really too short to allow those normal root processes to reestablish. But if you have an ingrowth period long enough like ours, you know, about 6 weeks, there's really no, this is the speculation or the agreement, I guess, that the actual installation of that root ingrowth core does not significantly affect the root growth or any of the root growth parameters if you allow that core to be installed or to sit in situ for long enough. There are parameters like, you know, in addition to the root injury piece, what's going on in that root ingrowth core can drive the root growth dynamics. So I'm not sure why my video is freaking out, but so whether or not the nutrient status is the same, so the nitrogen, whether or not the bulk density is the same, those kinds of physical soil parameters tend to have a larger effect on the ingrowth into those cores. And so we took a lot of care to, you know, collect soils within a week or so when we were going to be installing the cores, sieving them and trying to kind of mimic the natural soil conditions within our ingrowth cores. We took a lot of care to do that, to try and ensure that that root growth into the ingrowth cores would be as representative of field conditions as possible.
Thank you so much, Stella, and thank everyone for attending. This has been great. And this concludes phase 1 of the defense. So